Funding for Yale Cancer Answers is provided by Smilo Cancer Hospital. Welcome to Yale Cancer Answers with Dr. Anish Chagpar. Yale Cancer Answers features the latest information on cancer care by welcoming oncologists and specialists who are on the forefront of the battle to fight cancer. This week, it's a conversation about research into how cell communication can lead to cancer with Dr. Ben Turk. Dr. Turk is an associate professor of pharmacology at the Yale School of Medicine, where Dr. Chagpar is a professor of surgical oncology.
Ben, maybe we can start off by you telling us a little bit more about yourself and what it is you do.
Sure. So I was originally trained as a chemist, and it's been really a long-standing interest of mine to apply tools and techniques from chemistry to problems in biological and biomedical science. So the area that my research lab works in is the area of cellular signaling. And this is really about trying to understand how cells in the body communicate with other cells and also how they sense their environment. So a classic example of this would be how hormones work. So hormones are produced by a particular organ or tissue in the body, and then they travel throughout the body and they find a target cell to act on. And they work by binding to a receptor protein that's at the surface of the cell. And that binding basically triggers a cascade of events where a signal is transmitted from the outside of the cell to the inside of the cell and propagates within the cell to affect some change in the behavior of the cell or its physiology. And the way that I became interested in cell signaling to start with was based on its importance in cancer. So one of my first research experiences, actually before I started graduate school, was working on a tumor virus. And it turned out that the protein that I was working on acted by blocking the activity of a key enzyme that is involved in these signaling processes. And that enzyme has turned out to be a really important tumor suppressor gene.
For people who may be listening, they may be thinking, okay, I get it. You know, cells kind of talk to each other and there's this whole environment. And we've talked on this show about tumor microenvironments. But the concept of how these signals actually lead to cancer is still a bit mind-boggling because, you know, we know that in our normal state of being, we have signals, right? We have hormones, and those hormones do things like keeping our body in check, right? Keeping our metabolism the way it should be, growing the things that should be grown and not growing the things that shouldn't be grown, and so on and so forth. But it seems like it's really, when we talk about cancer, those processes that might be normal in some circumstances tend to go awry. And that's the part that gets a bit confusing. So can you talk a little bit more about how these signals kind of are different between cancer cells and normal cells?
Absolutely. So one of the important types of signaling molecule are proteins called growth factors. And under normal circumstances, whether a cell grows and divides or doesn't grow or divide is a very tightly regulated process. And so growth factors work kind of like hormones. So they bind to a receptor at the surface of the cell, and that transmits a signal that tells the cell to grow and enter the cell division cycle, that is to divide. So it turns out that in cancer cells, these growth signals are basically always in the on state, even if there's no growth factor present. And this can happen in a number of ways. Most commonly, this is through mutations in the very proteins that are involved in transmitting these signals. And so, for example, a particular mutation that one finds in a tumor might lock a key enzyme in an on state, and thus the cell will continue to grow and divide.
And so that then leads to two, I think, important questions. One is, why do they do that? And the second is, so how can you get them to change? How can you get them to realize that nobody's talking and that they don't need to divide?
Right. So why do they do it? That's a really important question. And really, you can think of a tumor as being sort of like a miniature evolving system. And so basically, a cells will acquire mutations, and if those mutations help the cell grow, help it survive, then those are going to be selected for, just like a natural selection evolutionary process. And then the other important thing, of course, is what we can do about it. And this sort of leads a little bit into the kind of work that my lab does. We study a particular type of enzyme that's really important for cell signaling, protein kinases. And these protein kinases, because they're very important in mediating these growth signals, also turn out to be important targets for anti-cancer drugs. Because if the kinase, for example, is always in its on state, and you can come up with a drug that blocks the activity of that kinase, then that can cause the tumor cell to stop growing or sometimes even to die.
But then it gets to the point of, the whole regulation. I mean, I think that when we think about biology, right, this whole on-off thing, it's not like the switches are always on or always off. There's this dance that goes on where, when they need to be on, they're on, and when they need to be off, they're off. And the body has this really miraculous kind of way of regulating the system so that things are exactly as they should be. And in kind of this wonderful kind of state of being. Whereas when cancers are always on, that's a problem. But then it also makes you wonder, well, if you give somebody a drug that turns that signal off, what happened to the whole regulation and the dance?
Right. Yeah. And so that's an important question, because these molecules that are important for regulating cells, protein kinases, other kinds of molecules that we're targeting with drugs, our normal cells have these molecules as well, non-cancerous cells. And so this is a big challenge in the field of cancer pharmacology, because you can have toxicities associated with these compounds that limit the amount of the drug that you can give to a patient, and thus limit their efficacy. And so this is one of these areas that's a real challenge in cancer drug discovery, is to make sure that you're not upsetting this sort of normal homeostasis, or as you put it, this dance that all these factors are participating in the context of normal physiology.
So when we talk about the work that's going on in your lab, and I'd love for you to tell our audience a little bit more in depth about what is the work that you're doing. When we think about protein kinases, I mean, it seems like these are enzymes that phosphorylate proteins. And there's lots of different proteins, and lots of different proteins get phosphorylated with these protein kinases. But when we think about cancer, I think a lot of times we start thinking about very specific cancers that come from very specific organ types, for example, and even the pathways are very specific. So there are general protein kinases that kind of have activity that seems to be more ubiquitous, and then very specific protein kinases. So can you talk a little bit more about the work that you're doing in your lab and which way you're kind of focusing and why?
Right, sure. So actually, that question of specificity is really the main core interest of my laboratory. So, there are actually a very large number of protein kinases. So humans have 500 genes for protein kinases. And of course, they're floating around within a cell that has thousands and thousands of proteins that they can potentially phosphorylate or tag with a phosphate group. And so that raises this question of, how does a kinase actually find just those proteins that it needs to phosphorylate in order to carry out its physiological function? And as you were suggesting, kinases can vary quite a bit in terms of how specific they are. But there aren't kinases that are completely non-selective that just phosphorylate everything. In order to have a sort of a functional, a specific effect, for example, of a growth factor binding to a growth factor receptor and sending a signal for the cell to divide, the kinases that are activated have to phosphorylate specifically proteins that are involved in that cell division process. And so a lot of what my lab is doing is really trying to understand where that specificity comes from. And it turns out that the specificity, by and large, is encoded in the actual amino acid sequences of the proteins themselves, specifically the amino acid sequences surrounding the site that is modified by phosphorylation. So proteins are made up of amino acids. They're basically strings of amino acids connected together. There are 20 different amino acids, and so amino acids are sort of the alphabet that make up proteins. And it turns out that the specific sequence of those amino acids is recognized by a protein kinase, and then that kinase puts the phosphate group on in the appropriate place. Now, the important thing from our perspective is that not all protein kinases recognize the same sequence. The various different protein kinases actually recognize different target sequences, and this is really where the specificity comes from. So you can think of the sequences within the proteins that become phosphorylated as basically specifying a code that directs a kinase to a single kinase and not other kinases to that protein for phosphorylation. And so what my lab has been trying to do really is to crack these codes. Basically, we have developed some years ago a method that allows us to quite readily identify the specific target sequence that is recognized by a particular protein kinase. And over the years, my group and our collaborators have actually applied this technique to basically every human kinase. We've profiled now almost 400 kinases. And this has given us a really rich data set that we can exploit in a number of different ways. So probably the primary way in which this data is important or useful is that it can help us figure out which kinases are phosphorylating which substrates. And this is important for understanding how a protein kinase impacts cell physiology, basically figuring out what the proteins are that it modifies and that it controls. And in order for those sorts of predictions to work really well, we have to have this information for all of the protein kinases, because basically what we can do is we can look at a particular site of phosphorylation and ask which of the 500 kinases that are made by humans is the best one for the job to phosphorylate that particular target sequence.
And so the story of how we can actually identify these areas and identify where those enzymes are reacting to those proteins comes to light. And presumably this gets us to how we can target them in terms of anti-cancer therapies. But in order to hear the rest of the story, we're going to have to just hang on for a very brief break as we take a medical minute. Please stay tuned to learn more about uncovering how cell communication can lead to cancer with my guest, Ben Turk.
Funding for Yale Cancer Answers comes from Smilow Cancer Hospital, where their screening and prevention program recommends that everyone perform monthly head-to-toe self-examination of their skin and protect themselves from the sun year-round. SmiloCancerHospital.org. Over 230,000 Americans will be diagnosed with lung cancer this year, and in Connecticut alone, there will be over 2,700 new cases. More than 85% of lung cancer diagnoses are related to smoking, and quitting, even after decades of use, can significantly reduce your risk of developing lung cancer. Each day, patients with lung cancer are surviving thanks to increased access to advanced therapies and specialized care. New treatment options and surgical techniques are giving lung cancer survivors more hope than they have ever had before. Clinical trials are currently underway at federally designated comprehensive cancer centers, such as the Battle 2 trial at Yale Cancer Center and Smilow Cancer Hospital, to learn if a drug or combination of drugs based on personal biomarkers can help to control non-small cell lung cancer. More information is available at yalecancercenter.org. You're listening to Connecticut Public Radio.
Welcome back to Yale Cancer Answers. This is Dr. Anees Chagpar, and I'm joined tonight by my guest, Dr. Ben Turk. We're talking about his research into uncovering how cell communication can lead to cancer. And right before the break, Ben was telling us about some work that's going on in his lab and how he and his collaborators have really unlocked nearly every kinase. A kinase is an enzyme that phosphorylates a protein, and that has a role to play in terms of how cancers develop. In fact, kinases control a lot of kind of biology. And so understanding where those kinases act is really important. So Ben, maybe you can pick up the conversation there and talk about how, now that you have this information, how do you put it into practice? How are you taking that information and really kind of moving the field forward in terms of understanding how cancer works, and perhaps even more importantly, developing targets for drug discovery?
Yeah, absolutely. So as you suggested, we have sort of two main areas that we're pursuing now with this data set that we have accumulated. And one of them is trying to understand basic tumor biology, or really even basic mechanisms of cell signaling as applied to other physiological or disease systems as well. And so I mentioned before the break that we can make use of the information we have to try to identify the proteins that are phosphorylated or modified by particular protein kinases. And these, of course, are the real players in the sort of processes that go awry in cancer. And so, for example, if we know a particular kinase is oncogenic, that is, it is important for causing cancer, then we really want to know what are the proteins that it acts on that are important for the development and maintenance of tumors. And so we have a number of projects in the lab that are really trying to understand that basic question of what are the specific targets of these kinases that are important for tumor biology. The other application, and this is something that we're involved with and that some of our collaborators are involved with as well, is looking a little bit more broadly at the entire collection of kinases as a whole, and trying to select from those kinases which ones might make good targets for future drug discovery. And so this involves using a specialized technique that's called phosphoproteomics that uses mass spectrometry information and instrumentation. And what that technique allows you to do is to map out the sites of phosphorylation on large scale. So something close to all of the sites that are phosphorylated on all proteins in a particular cell type. So how is this important? Well, kinase inhibitors are important anti-cancer drugs, but one of the limitations of kinase inhibitors and other sorts of targeted therapies is that they don't typically cure cancers. They usually slow tumor growth and delay progression for some amount of time. But what we'd really like to have are therapies that are really curative. And one of the ideas is that if we can find particular targets for therapies that one can combine with a kinase inhibitor drug, then that might make the activity of that drug or the efficacy of that drug more durable. And so in these phosphoproteomics experiments that I'm talking about, what you can do is you can take a cell that you're growing in a culture plate or even a tumor that you're growing in a mouse model, for example, and you can treat it with a particular kinase inhibitor drug. And then you can look at what phosphorylation sites go down and what phosphorylation sites go up when you treat with the drug. And the phosphorylation sites that go up are potentially mediated by kinases that are involved in these escape mechanisms, that is, that allow the cells to become resistant to the kinase inhibitor. And applying our data set, we can actually look at phosphoproteomics data and infer what the kinases are that are becoming upregulated or turned on when you treat with an inhibitor of a different protein kinase. And those kinases are potentially targets that could be used in these sort of combination therapies.
So it sounds like it's kind of like a 1-2 punch, right?
Yes.
You take out one kinase and find that phosphorylation goes up in another protein, and then you use a second kinase inhibitor to knock that one out. But just to play the devil's advocate, couldn't there then be a third one and a fourth one? I can understand how, the more punches that you can deliver to this tumor and the more durable the response is. But do we know how much more durability we get with the 1-2 punch? And is it potentially something to think about that you might need a 3 and a 4?
Right. And obviously, the more drugs that you add on, the more potential you have for toxicity and disrupting normal physiology that we were talking about in the first half. So this is a really active area of research right now. There are a lot of kinase inhibitor drugs that have been approved and that are in use now. And that means that there are a lot of different potential combinations that one can think about. And if you want to avoid this scenario of having to keep adding on more drugs and more drugs, you want to figure out which combinations are going to be the most effective. And because, like I said, there are a lot to choose from. And so I think prioritizing combinations that are being brought into the clinic in clinical trials is going to require a lot more sort of basic research and preclinical research being done in laboratories to really prioritize these combinations.
The word that you used was cure, which is something that a lot of patients aren't used to hearing when it comes to cancer. Can you talk a little bit more about maybe some initial results that you've seen in your work or things that you might be anticipating in terms of clinical trials that might actually lead to long-term durable cures for cancer?
Right. Yeah. So there's a lot of exciting work being done now, and a lot of things coming into clinical trials in the pipeline now that I think do raise hope for at least more durable responses, if not cures. So one area of research that is really brand new that I've been particularly excited about this year is the development of drugs that target a protein called RAS. Okay, and what's new this year is the development of what you would call pan-RAS inhibitors, and these have now entered early stage clinical trials. So I think you've talked about RAS on the program before. This is a key protein in these growth-controlling cell signaling pathways. It actually acts as an intermediary in between growth factor receptor kinases and the kinases that act downstream as the effectors in controlling cell proliferation. And for a variety of reasons, RAS has been a very difficult protein to develop drugs against. And about 10 years ago, the first RAS inhibitors were developed in laboratories and soon after entered clinical trials. And these were very exciting development, but there are some important limitations to those drugs. The main thing being that the RAS drugs that have been approved so far only target specific mutant forms of RAS. So RAS is one of the most frequently mutated genes in human cancer, and there are a number of different mutations at a couple of different sites that you find in different tumor types. What's exciting about these new compounds is that they will bind to and inhibit RAS independently of the specific mutation, and they'll also inhibit RAS that isn't mutated at all. And so this is significant because, first of all, it increases the spectrum of potential tumor types that one can treat with a RAS inhibitor, but also has the prospect of leading to more durable responses, because if some of these reactivation pathways or bypass pathways that are allowing cells to grow and divide, even when you suppress, for example, the mutant form of a RAS, a lot of these mechanisms actually depend on the wild type or non-mutant forms of RAS that are present in cells as well. So I think there is some hope there that this new class of compounds might, again, if not lead to cures, prolong the response further. But go ahead, sorry.
No, please.
So I was going to say another thing that I think is exciting about this particular class of compounds that is now entering the clinic is that they belong to a special class of molecules or special class of drugs that are called molecular glues. And these are drugs that basically work by sticking two proteins together. And what that allows you to do is target particular molecules that have been traditionally considered to be, as we say, undruggable. That is, they don't have good binding sites where a small molecule drug can interact, or they're not enzymes where the activity can be inhibited by a drug. And so I think from the standpoint of maybe a little bit further into the future, the fact that we have a designed molecular glue that is successful in inhibiting RAS sort of opens the door to designing more of these types of compounds to inhibit other targets that are important for cancer as well, and maybe even in other disease areas.
So I agree. That's really exciting. I wanted to get back, though, to the RAS story. One of the questions that people may be asking is, when you talk about these reactivation pathways that might be dependent on wild type RAS, people might be thinking, yeah, but don't my usual normal body functions also depend on wild type RAS? What happens if you target that at the same time?
Yeah, absolutely. That is a concern. And I think it's going to remain to be seen. These compounds are just entering human clinical trials now. The preclinical data, as far as I know, seem to indicate that they're well tolerated, at least in animal models. But you're absolutely right that there is a potential concern because unlike some of these other targeted therapies where you're really going after the mutated form of a kinase or the mutated form of RAS, here you are targeting a drug that may be involved and may be important for normal physiology. And in the context of cancer, one thing to think about is that these molecules that we've been discussing are important for cell growth and cell proliferation. And one context where that can be very important in actually fighting cancer is in anti-tumor immunology. So the cells of our immune system that people are now trying to exploit to target tumors, they have to be able to grow and divide too.
Dr. Ben Turk is an associate professor of pharmacology at the Yale School of Medicine. If you have questions, the address is canceranswers@yale.edu, and past editions of the program are available in audio and written form at yalecancercenter.org. We hope you'll join us next week to learn more about the fight against cancer. Funding for Yale Cancer Answers is provided by Smilow Cancer Hospital.